Good afternoon. Welcome to Lee County 4-H at Home. I'm Pam Curley and I'm glad to have you join us. Microbes have deoxyribonucleic acid, or DNA, and so do you. DNA is the genetic information that guides the development and functioning of all living things. Many aspects of genetic research require the extraction and isolation of DNA from cells of various organisms. In this lab, you will use household materials to extract DNA from a strawberry. What is this a picture or model of? Why do you think, if it is inside of every tiny cell in your body, that we can know what it looks like? Many organisms have similar DNA to other organisms, and it only takes a slight change in the DNA sequence to make a completely new organism. Today, you are going to experience how to extract DNA from a strawberry, and then I will encourage you to check out other fruits and vegetables in your kitchen so you can compare today's strawberry to other fruits and vegetable DNAs. So the first step to today's DNA extraction activity is to put the strawberry into the plastic bag and carefully mush it up until it is the consistency of baby food. All right, so while you are doing this, you want to be careful not to put a hole in the bag. Also, you can let all the air out. It makes it a little easier to mash the strawberries. The purpose of mashing the strawberry is to rupture the cell membranes and to release the molecules contained within the cell and within its organelles. Measure 2 tablespoons of detergent into the bag. Measure a half a teaspoon of salt into the bag. Mix the detergent and salt into the fruit and vegetable mixture to make sure it is thoroughly spread throughout. Measure a half a cup of warm water into the plastic zip bag. Close the bag carefully and mix the water thoroughly with the other contents. Now allow the mixture to sit for 5 to 10 minutes to allow the contents to fully combine. Place the coffee filter inside of the funnel and place the funnel over the top of the container. Carefully pour the mixture from the bag into the funnel. The coffee filter will only allow the liquid portion to go into the cup. Using your fingers, carefully mash the mixture caught by the filter to make sure all the liquid drips into the cup. Remove the funnel and dispose of the coffee filter. Carefully place the filtered solution into the test tube until it is a third full. Now cool the mixture in your ice water bath for 5 minutes. Slowly add cold alcohol to the test tube, filling it 3/4 of the way full. Hold the test tube at an angle when you're adding the alcohol. See the bubbles forming? You will now see the DNA precipitate or float out of the solution near the top. It will appear as a clear, goopy substance. Here is a picture. Scientists have figured out ways to extract DNA from organisms so they can compare their genetic information to other living things. This is how we have been able to learn a great deal about organisms that lived many years ago, and also find modern-day organisms that they are closely related to. Today's 4-H at Home is a part of our Lee County Still Growing Agricultural Series, highlighting strawberries. There are a lot of great local farms with strawberries available now, so be sure and download the Visit NC Farms app to find a producer near you. This lesson is a small portion of the NC 4-H microbiology curriculum. If you are interested in using this in your classroom, contact me at NC Cooperative Extension, Lee County Center.